Hi, my name is Grace Caputh and I'm a sophomore at Hendrix College. In this video, I'll be walking you through the process I took to relabel, barcode, and repackage in archival quality paper a collection of bryophytes to better preserve them in our college herbarium. I also digitized the label information, which is available on our herbarium database through CERNEC, the Southeast Regional Network of Expertise in Collections. In 1949, Dr. Tom Clark of Hendrix College collected over 150 samples of bryophytes with the help of Dr. Charles Arzini of the University of Michigan. Bryophytes is a term used to denote a group of plants that consists of three categories of non-vascular land plants: hornworts, liverworts, and mosses. Dr. Clark's collection includes two of the three categories, liverworts and mosses. He collected in the northern Michigan area, mostly from Sheboygan, Emmett, and Mackinac counties, though we also found several specimens from Sanik Island of the Aleutian Islands. He stored the samples in small paper packets and labeled them with the determined scientific name, place of collection, and other details about the location and habitat of the bryophytes. The packets have been kept in these shoeboxes for over 70 years when they were discovered in the college herbarium. Here are some of the original packets. As you can see, Dr. Clark made notes on the outside of the packet. On the inside, there are preserved samples of moss. Occasionally, he made morphological notes on the inside of the packet. There are also a few where he went back and corrected his original determined nomenclature as designated by the X here and the name rewritten in pencil below. This is one of the samples from the Aleutian Islands. This sample was collected by Sabi R. West and determined by C.B. Arzini. It was collected in December of 1949. Below. In total, there are 38 liverwort specimens and 115 moss specimens. We have mosses from 38 different families and liverworts from 22 different families. However, there are no hornworts in this collection. Now you may be wondering why we bothered to keep around these old moss samples, and even why we need an herbarium at all. Herbaria house collections of plant samples that are key sources of information for research in fields such as ecology, anatomy, taxonomy, conservation biology, and many more. These samples are used as comparative material in research, and though they seem old, through meticulous drying, pressing, and preservation, these samples are still well intact and valuable. Herbarium material can be used to make chemical and DNA analyses, plant illustrations, morphological measurements, pollination studies, and much, much more. The information we collect and store in herbaria is also important for creating and updating online databases. This is the CERNEC database. CERNEC compiles data and images of specimens from over 233 different herbaria from the 14 southeastern states. Here we have digitized and uploaded all of the information and labels from each of Dr. Clark's specimens. Although major pushes are being made to digitize botanical and biological collections, mosses and other bryophytes are usually woefully underrepresented, so it was important that we are able to database ours. To begin the process of databasing and repackaging the samples, I first went through each packet and recorded the information by hand in a spreadsheet. During this stage of the process, I needed to compare Dr. Clark's determined scientific name to the current accepted taxonomy. To do this, I compared the original name to three different sources: the Plant List, the International Plant Names Index, and Dr. Howard Crumb's Mosses of the Great Lakes Forest. Mosses of the Great Lakes Forest is actually a compilation of Bryophyte species from the same areas of Michigan in which Dr. Clark collected samples. The book was published specifically for students at the University of Michigan Biological Station, from where Dr. Clark's colleague, Dr. Charles Arzini, was graduating at the time. This book had an account of most of the moss samples from Clark's collection and was invaluable in determining the current taxonomy of many of them. To determine the current taxonomy of the moss samples, I turned first to the Mosses book, then to the other sources where the Moss book was unclear. Once I finished recording the data from each packet into one big spreadsheet, we printed new labels on archival quality paper to better preserve the packets and specimens from damage or deterioration. In order to print each label, we transferred the spreadsheet data into a template in Word. The label features the current taxonomy first and foremost, and Dr. Clark's original determined nomenclature is printed at the bottom. As you can see, the new label was printed with set margins for folding purposes. Here is a quick demonstration of how I folded the moss packets. Using a regular sheet of copy paper as the template for folding, first, you fold the top and bottom thirds of the sheet. Next, you fold the two sides of the packet. Finally, to create a sealed pouch for your moss sample, fold in the corners where the bottom third and side folds come together. This is the base of your moss packet. Next, we take a printed sticker barcode and place it on the bottom right-hand corner. The barcodes are used for easy lookup in our Herbarium database. Now your packet is complete. After folding and barcoding all of the new packets, the moss samples will be transferred to the new packets, organized by their botanical family, genus, and species. I will also cut out the original labels and store them inside the new packets with the moss samples. From there, the packets will be restored in the herbarium and the project will be complete. The mosses will be accessible for future research and study, along with about 6,500 other plant, algae, and fungi specimens in the Hendrix College Herbarium and through the CERNEC database. In the future, we hope to image the specimens using a lightbox and a camera at the Arkansas Natural Heritage Commission. Thank you for watching, and I hope you enjoyed this presentation.